Welcome to today's deep dive, everyone. I'm so glad you could join us because today's mission is, well, it's honestly mind-blowing. We're unpacking a brand new paper from Nature, and it's titled Competing Programs Shape Cortical Sensorimotor-to-Association Axis Development.
Yeah, it's a bit of a mouthful, but the concepts inside are just fascinating. It really challenges everything we thought we knew.
Totally. Because, for decades, neuroscientists have understood how the brain processes basic senses, like touching a hot stove. But the areas responsible for complex, abstract human thought, how those develop has basically been a massive mystery.
Exactly. And today we're looking at a brand new framework called the mind model. It basically reveals that our brains are built through this epic microscopic turf war while we're still in the womb.
A turf war in a fetus. It's wild to think about. And our goal today is to help you understand not just what makes up your brain's geography, but why it's organized that way, right? And how it links to evolution and conditions like autism.
Right. It's about getting to the why of our brain structure. To start, we have to look at the fundamental map of the cerebral cortex, which researchers call the sensorimotor-to-association axis, or just the SA axis.
Okay, SA axis, so break that down for us.
Sure. So at one end of this axis, you have the sensorimotor areas. These are the primary areas. They are unimodal, meaning they handle very specific, concrete tasks, like processing direct sensory input from your eyes or ears.
Or like sending a direct signal to move your arm. Oh, yeah.
Exactly. Very localized, very direct. Now, at the completely opposite end of this axis, you have the association areas. These are transmodal.
Transmodal meaning they kind of mix things together.
Right. They are these distributed networks in the prefrontal, parietal, and temporal cortices. They don't take in direct senses. Instead, they integrate all that information to support, complex abstract cognition.
The paper actually uses this incredibly vivid visual analogy that I love. Topographically, if you look at the brain, it looks like these highly specialized primary sensorimotor areas are just focal islands.
Yeah, islands of concrete processing.
Right, floating within this vast interconnected ocean of association cortex. The islands handle the raw data, and the ocean does the deep thinking.
It's a great analogy. And the mystery here is that we've actually understood how those primary islands form for a long time.
Because they're driven by the thalamus, right?
Yes, exactly. They're driven by what we call first-order thalamocortical inputs. Basically, sensory signals from the thalamus act as anchors for these islands. But the real question is, how does that vast ocean of association cortex develop?
Right, because how does it know to just go around the islands? Wait, actually, if the brain starts out as just this undifferentiated mass of cells in a fetus, how do these areas know to separate it all? Like, why doesn't it just overlap into chaotic static?
That is the exact question this paper answers. If they just grew into each other, you'd have a non-functional brain. There has to be an active mechanism keeping them apart.
Okay, so let's do a quick subtopic summary for you listening. Basically, your brain is divided into sensory islands and an association ocean along this SA axis. And I want to pose a question to, think about how you're listening to this right now. Your primary areas are hearing the actual sound of my voice, but your association areas are connecting it to your memories and understanding the words. Which network do you think dominates your daily life?
It's a fun thing to consider. And to answer your previous question about how they avoid overlapping, we have to introduce the mind model.
Yes, the mind model, which stands for multinodal induction exclusion in network development.
Which is a very fancy way of describing that fetal turf war we mentioned earlier. This model is a new conceptual framework that explains how that SA axis develops.
So what's actually happening in the brain during this time?
Well, it happens during early to mid-fetal development, roughly periods 3 to 7. Brain development is essentially driven by two opposing genetic programs that are quite literally competing for space.
Okay, so who are the fighters in this turf war?
First, we have the pericentral program. This one emerges around the frontotemporal poles, so the front and sides of the developing brain. And it progresses inwards toward the center of the undifferentiated cortex.
And this one is building the ocean, right? The higher order association features.
Exactly. It's sweeping in from the outside. But then you have the central program. This one is induced right in the center of the brain by those sensory signals from the thalamus.
The anchors for the islands.
Right. And it's establishing those primary sensorimotor areas.
To help you remember this, we came up with a little mnemonic. Think PPP and CCC. So for the first one, PPP is pericentral, which starts at the poles and handles philosophy like your complex thought and association.
I like that.
Right. And then CCC is central, starts at the core and handles concrete senses, those primary islands.
That's a perfect way to visualize it. But the key to the mind model is the mechanism of exclusion, because the central program doesn't just passively sit there.
It fights back.
It fights back hard. As that pericentral program advances toward the center from the poles, the central program aggressively excludes it. It pushes back. They fight for space.
Oh, wow. So the islands are actively keeping the ocean at bay.
Exactly. The central program secures its territory by rejecting the pericentral program.
Okay, let's do a quick 5-minute recap and bullet summary of what we've covered so far.
Okay.
Brains have primary islands for basic senses and vast association oceans for abstract thought.
Right.
Bullet 2. The Mind-E model explains how these areas form through a literal microscopic competition.
Right.
And bullet 3. The pericentral programs sweep in from the poles to build the ocean, while the central programs push back from the core to secure those sensory islands.
That sums it up perfectly.
So a quick question for you listening at home. If your brain is basically a map being drawn by two competing artists, what happens when they actually meet at the borders? What does that boundary look like?
Well, to see what happens at the border, the researchers had to dive deep into the molecular evidence. They used something called principal component analysis, or PCA, on fetal brains.
Wait, PCA. Can you explain what that actually shows in this context?
Sure. PCA is a way to find the biggest patterns in huge data sets. Early in development, the PCA showed that gene expression is just polarized physically along a front-to-back axis, but by late fetal development, that completely shifts.
Oh, so it stops being just physical geography.
Exactly. It shifts to perfectly match the functional SA axis. It's no longer just front versus back, it's island versus ocean.
That's incredible. So the brain flips a switch and says, okay, enough scaffolding, let's draw the actual borders.
Right. And in that transcriptomic data, they found the two main molecular warriors enforcing these borders. Two specific genes.
So let's name these molecular warriors. We've got SEMA7, which is the central program gene highly expressed in the sensory islands. And then we have PLXNC1, which is the pericentral gene, enriched in the frontal and temporal association ocean.
Yes. And the mechanics of how they interact is it's fascinating. SEMA7A acts as a ligand, and PLXNC1 is its receptor.
Normally, don't those two things bind together to send a signal?
They do, but in this specific context, when they meet, they actively repel each other. It is aggressive axonal repulsion.
Wait, really? The researchers actually saw them repelling each other?
They did. They proved this using an ex vivo culturing strategy. Basically, they took fetal neurons that express PLXNC1, the association ocean neurons, and grew them in a dish.
Okay.
Then they introduced primary motor neurons that express SEMA7A directly into their path. When the growing axons of the association neurons encountered the motor neurons, they literally repulsed each other. They stopped growing and turned away.
Oh, wow. It's kind of like if you take two strong magnets and try to push the identical positive ends together. They just physically push away from territories where they don't belong.
That's exactly what it's like. This magnetic repulsion creates a graded transition. It prevents the networks from getting tangled and establishes the correct connectivity across the brain.
Okay, it's time for another subtopic summary. We basically have these molecular magnets, SEMA7A and PLXNC1, acting as border patrol to keep the islands and oceans separate. And for you listening, just think about this. We literally have microscopic chemical forces pushing and pulling inside our heads to keep our thoughts clear. Can you imagine the precision required for billions of neurons to find their exact place using just chemical repulsion?
It is a staggering level of biological precision.
It really is. But it makes me wonder, is this turf war just a human thing since our association ocean is so massive?
Actually, no, and that's where the evolutionary magic comes in. This mechanism is highly conserved. They found the exact same SEMA7A and PLXNC1 boundaries in macaques and mice.
Wait, mice have this too?
Yes, even marsupials like opossums and avians like chickens, the basic blueprint is ancient.
So if a chicken uses the same molecular border patrol, what makes the human brain so different?
That is where it gets really interesting. While the mechanism is the same, human transmodal association cortices have undergone a massive evolutionary expansion. Our ocean is huge.
Right. And crucially, they exhibit something called neoteny. Neoteny, like remaining young.
Essentially, yes. It means a prolonged, delayed postnatal development. Our association networks stay plastic and continue wiring themselves for a very, very long time after we're born.
So we basically keep building our brains ocean way into childhood and adolescence, which explains why we can learn so much and have such complex language.
Exactly. It's our greatest cognitive gift. But there is a dark side to this expansion, a cost to all this complexity.
Yeah, the paper talked about this. When they looked at the gene modules that build this association cortex, they found they share features with the allocortex, things like the amygdala and hippocampus.
Right, very ancient emotional structures. And these expanding regions are densely packed with WNT signaling genes and genes for complex synapse assembly.
And crucially, they are packed with autism spectrum disorder risk genes.
Yes. That's the core clinical vulnerability. The exact same lengthy, complex developmental window that gives humans our advanced abstract cognition also creates a massive surface area vulnerability.
Like, the longer you spend building a really complicated house, the more chances there are for the wiring to get a little crossed.
Precisely. It creates a huge window of risk for neuropsychiatric and neurodevelopmental conditions like autism. It's an evolutionary trade-off.
Wow. Okay, let's do our final rapid-fire recap for you guys.
Yep.
Bullet 1, SEMA7A and PLXNC1 are the specific genes that act like those repelling magnets to draw the brain's borders.
Yep.
Bullet 2, this biological blueprint isn't just human. It's ancient, shared with mice and chickens. And bullet 3, human association areas grow much larger and mature slower, which unlocks our high intelligence, but also introduces neurodevelopmental risks like autism.
Perfectly summarized.
So my question for you listening, does knowing that our greatest cognitive gifts are inherently tied to our deepest neurological vulnerabilities change how you view human intelligence? It really makes you think.
It fundamentally reframes how we view neurodivergence, honestly. It's all part of the same complex mechanism.
It really is. Bringing it all back, every time you seamlessly combine the sound of a voice using your primary auditory island with the emotional memory of a friend in your association ocean, you are using a network forged by this microscopic fetal turf war.
And it leaves us with such a fascinating, provocative thought to build on.
Yeah, what's that?
Well, if the precise borders of our sensory and association areas are defined by this competitive push and pull of the mind model, what happens if that exact balance shifts just slightly during early development?
Oh, wow. Like if one program is just a tiny bit stronger.
Exactly. Could a slightly stronger central program explain why some people have hyperacute sensory talents, like perfect pitch?
Wait, and then on the flip side, what if the pericentral program is stronger? Does that create individuals prone to deep abstract daydreaming because their association ocean is literally pushing further in?
It's highly possible. The architecture of our perception might just be the result of who won a microscopic battle by a fraction of a millimeter.
That is just incredible to think about. Thank you all so much for joining us on this deep dive. Keep questioning the world inside your own head, and we'll catch you next time.